everybody, I'm Tall.
And I'm Small.
And today we are going to continue our preparation for the MCAT by going through AAMC's actual study guide and using content from all of our different third-party sources to expand on that study guide and learn exactly what's going to be on the MCAT. We are going through Psych right now, and Small is going to be teaching us about vision and the eye.
I bet you didn't see that coming. Hey, I said the punchline right this time.
So,
this is our third time starting this video, in case anyone was wondering.
Don't break the fourth wall! Okay, so we are going over the eye.
Yeah, okay. And I wanted to mention something that I had never known before. Actually, let's start this way. So a lot of people, I think, learn it at a very surface level for what they need to in whatever basic bio class they take. And then they get to physiology and they're like, oh, wow, there's so many more layers to this, quite literally, than I thought. Like, the eye has 3 layers, and I never knew that, or paid enough attention, I guess, or encoded it as well as I should have. So,
another little hint for everybody, use words like encoding so that you get meta with your MCAT studying.
yes.
That didn't happen.
Yeah.
Anyway, continue.
Yeah, okay. So, let's start with the outermost layer and really corny jokes. That was a pun. That was unintended. Just stick with it. Okay.
Cornea.
Haha.
So what is that?
So, I'm getting there. Don't steal my thunder. So the most outer layer is your sclera. It's the white of your eye. And it covers all the way around the eye except for where the optic nerve leaves in the very back and the very front of the eye, which you see out of, which we would like to be clear and not white.
Right.
And so the very front of the eye is called the
Cornea.
And it would get scratched if somebody threw a piece of corn at your face.
It definitely would.
That is how I remember cornea. So that, I think, basically takes care of the very outermost layer.
So it's sclera.
Yeah.
Whichever way you pronounce
there's no H.
it. The sclera then is the white part, which goes all the way up to the cornea, which is the entire outer part covering the colored part of your eye?
Yeah.
Okay.
All of the clear part on the very outside. I
Gotcha.
think there's actually a very thin layer of cells all the way across your eye. But yeah.
Which would get scratched if someone threw corn at you.
Right, over that cornea.
Understood.
Ba-dum-bum. You gotta get cheesy, guys, to remember it.
That's the best way to remember things.
The very middle layer is called the choroid, which is kind of hard to say, but it has all of the major function of the eye other than signal transduction. So what that means is there are some dilator pupillae and some constrictor pupillae. That word is hard to say. And so if there was a bear running at me, what nervous system would be stimulated?
Probably your sympathetic.
Right, and then what would happen to my eye?
It would dilate to let more light in.
Exactly, because I need to get going, right? Because this bear is going to eat me.
So the choroid is like your pupil. It's like behind the very most outer layer, but it's not back in the retina. That's what I was thinking.
Right. It's behind your sclera, but in front of it. Well,
does it include the lens and all that stuff? I know we're about to get into that. It doesn't.
Well, yeah, I don't think it includes the lens, but it is between the sclera and the retina. And it's in charge of the muscles that allow for pupillary constriction and dilation, and it's continuous with the iris, the really pretty colored one, part of the eye that people like to sing about in songs. It also contains this little not muscle, but it's almost a gland, because it is what's responsible for giving you your aqueous humor in the anterior section of your eye.
Okay.
And it also contains the thing that drains the aqueous humor.
Interesting.
So anyway, the middle part of your eye is really important for basic function throughout the day, other than signal transduction, because that is reserved for the retina.
Okay, so it also contains those muscles that then would dilate or constrict the pupil.
So we talked about dilation being with, if I'm sympathetically stimulated, if there's a bear running at me, which I'm going to use every single time because I love that example. So when I'm in a really boring lecture, maybe, none of my teachers, of course, but if I were to ever be sitting in a boring lecture, what would happen to my pupils, and what would be stimulated to make that happen?
They would constrict, probably due to parasympathetic, which is your body telling you this lecture sucks. Let's go bed.
Let's go digest.
Let's rest and digest. Poop on the floor and go to bed.
Negative. Social cues. We'll get into sociology later.
So if a cute puppy was running at you,
yeah,
would your pupils constrict?
I think at first they would be sympathetically stimulated because you'd be super excited about this little puppy, right?
Maybe, yeah.
Says the girl with a tattoo of a puppy. So, anyway, so we talked about the outermost layer, the middlemost layer. I don't know if you can be the most middle, but the middlest layer. Yeah, like the middlest brother. And so, kind of in the very middle of the eye, there's that anterior section we said that has an aqueous humor. You can remember that with anterior aqueous.
That's like a big chamber almost, right?
Yes, and it is separated from the vitreous humor, or the back posterior part, which is actually bigger and has a gel-like substance that kind of helps our eye keep shape. And it's separated by the lens, right? The crystalline lens is kind of in the very middle of your eye. And it is it's like a crystalline structure.
So if we're talking from front to back, because that's how a lot of people like think about the eye, we have, as of right now, the excluding the sclera on the outside. We have the cornea. Then we have, is it the aqueous humor? Because that's like the black pupil of our eye. That's actually just liquid. That's the aqueous humor, right?
I don't think it's colored at all. I think it's very clear. I think the black you see is literally the optic nerve.
Right, yeah. That's what I meant, but like...
Oh, no, it's like the back of the retina, right? You can see it, but it looks black or something.
Yeah, I think so. But what we're looking at as the black of our eyes, once we get past the cornea, we are then going into the aqueous humor. Yeah. And then right behind the aqueous humor, which we know can be expanded or contracted using the choroid, which is the middle part of the eye with those constrictor muscles. Once we get behind that, then we are at the lens.
Yeah, once you go through the aqueous humor, you hit the lens, and then you go through the vitreous humor, and then you get to the...
retina.
Which is amazing, right?
Very important.
And it controls, or it holds, all of our what kind of receptors?
Photoreceptors.
Right. And so if I were to say the words cone and rod to you, go ahead and give me some brainstorming. What do you think of when you hear cones and rods?
Cones. I think of orange and white traffic cones. I think of those little like multicolored cones that they would have us do in PE as a kid. Are you expecting me to cut you off
at some point?
No, I'm just thinking of cones. Thinking of ice cream cones. I'm thinking of Neapolitan ice cream cones. I'm kind of giving away all of my mnemonics for cone.
Okay, in the context of a photoreceptor, when I say cones, what do you think of?
Colors.
Good. Okay. And do we have more cones or rods? Do you remember?
We have way more rods, right?
Way more rods. We have like 120 million rods compared to 6 million cones. And where are those cones distributed?
The cones are mostly right around the fovea, right? Is that...
So they're highly concentrated in the very center of our vision, which means that we can see detail and color way better if it's smack in front of our faces.
Right.
And the Fovea Centralis is a little dimple in the very back of our retina where our sight is the best it can be. It's almost entirely concentrated with cones, and so it's so saturated within that our vision is like perfect there, or almost perfect.
That's because of the way that light bends. And once again, I'm about to prep everybody. What we are about to talk about in little detail, you should go through from the physics side, because it's almost guaranteed to be a physics question on the MCAT.
It's actually, I think, highlighted on the AAMC outline.
Oh yeah, they basically like guarantee you there's going to be a passage.
We're going to test you on this.
But that fovea is really dependent on light going through the lens and then moving through the vitreous humor correctly, and then focusing, like, you know, the two basically beams of light in a triangle kind of like coming to that point right on the fovea.
Yeah. Yep, exactly. And so if I'm nearsighted, do you know the technical term for nearsightedness? That
means that the light converges in front of the fovea, right?
Yep.
I don't know the technical term.
It is myopia.
Myopia.
Yeah, myopia, nearsightedness. And you're right, the two light rays kind of converge before they reach the retina and that fovea. And so then you need a concave lens to be your glasses, kind of be an artificial lens to help direct those light rays to converge back on the retina as we need them to.
Okay, concave lens if you are if you are nearsighted.
Yes.
Because concave will push that focus point back.
Yes, and so the way I remember that is actually going into a psychology thing that I read about a couple days ago, and that's when you drink alcohol, you actually, it's kind of a behavioral version, but you're nearsighted in that you don't remember that there's consequences to your actions, so you're nearsighted as far as your actions go.
Okay.
And we, this is going to sound terrible, but like, if an alcoholic could be put in a cave where he couldn't hurt himself or do anything bad to anyone else.
They call it prison.
Oh, no. That is how I'm going to think of myopia being nearsightedness.
Oh. Okay, yeah,
But once you hear that term often enough, you no longer have to rely on that semantic memory game to remember it.
Mm-hmm.
It will just be a part of your crystallized memory.
Oh, I cannot wait for us to start talking about memory.
Yes. So,
okay.
And it'll be in your long term. So, yeah.
Myopia, nearsightedness. Do you happen to know the farsightedness? Or
yeah, so it would just be the opposite. So with a lot of these things, if you can remember one of them, you just know that the other one is the opposite, which we use in a lot of things. Mm-hmm. Like how the optic tract works on one side, works in the exact opposite side on the other.
Right.
And the exact opposite way on the other side.
Right.
But yeah, so for farsightedness, it's hyperopia is the technical term for it.
Hyperopia. Oh.
And so those light rays would converge past the retina.
Yes.
So it still doesn't reach the fovea.
Mm-hmm.
And you would need a convex lens to fix that.
Concave, because you put them in a cave if you're a myopic alcoholic,
Mm-hmm.
Which would push it back. And then it would be convex for, which, it's just the opposite. If you can remember one, you can remember the other. Convex, if you are farsighted and you need that focus point to be shifted forward a little bit on your fovea.
And now I don't really know the physics behind it. It's something I'm going to have to read about, but we know at least that much for right now.
Mm-hmm.
Good starting point.
Yeah.
So I think we were back on cones and rods.
Yes.
And so we kind of in our first video started talking about how exactly they transduce signals, and I don't think we need to know that. I don't think the MCAT wants us to know exactly how the changing of retinol does what it does and all that. So anyway, we do need to know, however, I think we're through all of this. Oh, one thing we do need to talk about for the retina is that there is a hole in it, right? On both eyes. Do you remember what that is?
Oh, geez. Well, it's where the nervous stuff starts to happen.
Yep, where the optic nerve leaves the eye, we actually have a hole in our visions. We have a blind spot, and we have two of them.
Oh, that's right.
But because we have two eyes, remember what happens?
It bypasses that blind... Oh, I know it bypasses the blind spot, right?
Yeah, the sight of one eye will... there's enough overlap to where we don't see any of the blind spots. But maybe if something happened to one of our eyes, we would have a hard time seeing anything in that area if we only had, monocular vision.
So if I covered one of my eyes for long enough, there might actually be a spot in my peripheral vision or even just in my normal vision where if I could look in that spot, I wouldn't see things.
Yeah.
Whoa.
Yeah, so that's very cool. And so we talked about cones being heavily concentrated in the center, and they help us see color and little details. And there's way less of them, super specialized.
Yes.
And then rods actually work better in the dark, and their purpose in the light is kind of to help us detect that there is an object there, even though we might not be able to see it real detailed. So, and they're higher concentrated in our periphery.
Yes.
So in physiology, we did a lab where they would take a long piece of yarn, and they would hold one side on the very top of someone's head, and then someone else would take 3 different colored circles, and they would pick one of them and put it on the other end of the string. And they would start from behind the person, and they would start going around in a circle following the radius or the circumference of the string, basically. And you would have to mark down exactly when that person started to see an object around the side of their head, and that would be due to their rods. And you would ask them if they could tell the color, and they would always say no, you can't tell it from that point. But once it got in front of them enough, their cones had enough power at that position to say, oh yeah, that one's blue, or that one's green, or that one's red.
Something interesting, it's kind of a weird thing, but I remember my entire life growing up, I always, I guess I like to just focus on something because I could always, I knew instinctively ooh, I can do something cool if I just look at this one object. Normally it has to be a colored object. If I just stare at it intensely enough and I just keep staring at it, almost in a meditative way, and I just keep looking at this object, eventually I can notice that things in my periphery, like you're wearing a blue shirt, but if I'm staring at that thing, then eventually if I stare hard enough and I just focus on that one thing, I can actually tell that everything in my periphery starts to lose color and go gray.
Oh, wow.
Basically. I don't know if anyone else has ever had that that experience. And then when you look out, you get like that like hypersensitivity stuff where everything is like a crazy color kind of in your periphery.
I think that desensitization.
Yes.
I think we've had a passage on that before. I wonder if that's a version of that, maybe.
Yeah, it might be.
Where your periphery gets affected. So one more time, we talked about this already. Well, actually, we didn't talk about after a signal hits the photoreceptors. We didn't talk about how that signal gets transduced.
Yes, goes through the optic nerves.
Right. So the transduction, that creation of that transition from, this stimulus is a light ray.
Mm-hmm.
How do we get it into an electrical signal? We said that happens at the photoreceptors.
Yes.
And so what is the next cell after that in the retina?
Photoreceptors, I know we have ganglion cells, we have amacrine cells, we have bipolar cells. I'm going to say it's bipolar.
Bipolar is right. And you can kind of remember that because it is a connective cell.
Mm-hmm.
It's got axon termini that butt up to the photoreceptors,
mm-hmm.
and then it's got its cell body, and then it's got another axon kind of piece and axon termini that go to the ganglion cells.
Yeah, so it like literally is bipolar.
Yeah.
In my head, the thing that came to mind, I don't know if this will help anyone else, is carbon dioxide, because if you like look at only 2/3 of carbon dioxide, it's a C and an O, that's polar.
Yeah.
And if you look at the other half, it's a C and an O, that's polar. But altogether, it's not polar.
We're getting really meta up in here.
Bipolar.
Whatever helps you remember that.
Yeah,
so yeah. So photoreceptors, bipolar cells, and then we're just going to do the very basic pathway, and then we'll talk about the other fancy cells.
Okay.
But after bipolar, there's the ganglion cells.
Mm-hmm.
And these are the ones that actually converge into one nerve to form the optic nerve from both eyes. And I remember that because a gang is a group of people that acts with one goal in mind.
Yeah.
Yeah. So they're
like
unified or whatever.
Mm-hmm.
So it goes photoreceptor, bipolar cells, ganglion cells. And then the ones that we mentioned, the amacrine, and what's the other one?
I don't know the other one, but I do know amacrine.
Yeah, amacrine and something else helps us kind of shore up the fine details. But as long as you know the specific ones, then I think you can just know that those help.
Probably not going to be too much on the amacrine and stuff like that.
Probably not very high yield. So,
fine dining and breathing.
yes, exactly. To kind of wrap this one up, let's talk about the visual pathway. So you walk me through this pathway.
Okay. So the visual pathway, this is probably where most of the questions are going to come. Well, not most, but this is where a lot of questions, high yield questions, are going to come from. So this is where people say that like your brain flips images. And,
no, not this part yet.
No, not like upside down. I mean like it crosses over through the. Well, let's walk through it.
Yeah.
Okay, so your vision is kind of divided by your brain into right and left. In each eye.
Oh, you're getting into that. I wasn't even talking about that yet.
What are you talking about?
Just for as far as we've gotten so far.
Okay, so
to the optic nerve, and then I will let you totally take over for that part.
I was putting it in high gear or low gear. I'm not sure which one goes faster because I don't know how cars work, but I ride a bike. So we go from let's do this all. So we have the cornea, that's right. The cornea. Light goes into the cornea through the aqueous humor, then goes through the lens, which then starts to focus the light through the vitreous humor, which is the giant cavern in the middle of your eye, which hopefully will then focus that light on the dimple, which is the fovea. The fovea contains a bunch of cones and some rods, and those will activate, which we don't probably need to know much about that, except for maybe like retinol is the thing that gets triggered. Which will then start a cascade of electrical signal through bipolar cells. The bipolar cells will then connect to ganglion cells. The ganglion cells all kind of bundle up together to form the optic nerve.
Beautifully done.
Yes.
So then, do you want to teach this part about the double hemisphere kind of thing going on?
I'm going to give it my memory shot right now, and I want you to correct me along the way.
Sounds good.
So, vision's divided up in the left and right.
Yep.
So, in that optic nerve, we have, it's like two parts, right? Like, because it's divided up into the left and the right, everything, so if I'm looking at my right eye, everything in the left half of my right eye gets sent into one part of the optic nerve. Everything in the right half of my right eye gets sent into another part.
Yeah, and this is where it gets really confusing to teach other people, even if you've got a good hang of it. Like, sometimes just to start talking without having a picture in front of you is really hard. And so one of our friends that we study with actually came up with 2 simple rules to keep in your head so that you never have to kind of start from scratch and work through and try to memorize all these different parts to it. So just keep in your mind that everything on your right field of vision is sent and processed to your left hemisphere. And everything in your left field of vision, so that includes your the temporal side of your left eye and your nasal side of your right eye gets sent to the right hemisphere. So that's the first rule. And then, that there's a nasal, a medial, and a temporal outside side to our visions. So we mentioned both of that.
Okay. So, I see what you mean. Yeah. Okay, so on my right eye again, let's go with the right side of my right eye.
Your temporal side of your right eye.
Yes, temporal side of my right eye. That is eventually going to end up, we'll talk about the path, but that's eventually going to end up in the left side of my occipital lobe.
So what is the other eye that has a right side?
What? The left eye has a right side.
Right, and that's on the nasal part of the left eye, right?
So the nasal part of the left eye and the temporal part of the right eye are both going to get sent to the left side of the occipital lobe.
Yeah.
Because they're the right field of vision in each eye.
Exactly. Beautifully said. And so physiology teachers teach it in a couple of different ways. So by the time people get to the MCAT, they've heard it in a couple of different ways. And as long as you keep that one simple rule straight that Alec made up, and so I'll repeat that one more time because I think it's really important and helpful. The left field of vision in both eyes is controlled by the right hemisphere, and the right field of vision in both eyes is controlled by the left hemisphere.
So this brings up this crossing over thing that people always think about.
Yeah.
It's not necessarily that all of your vision gets crossed over.
Right.
Because if we know that our right field of vision is getting sent to the left part of the brain, then you have a left eye that has a right field of vision. So that nasal part of your left eye will actually, it'll kind of go to the middle, but it basically just goes straight back to the left. Whereas the right field of vision in your right eye will actually have to cross, that optic nerve will have to cross in what I think is called the optic chiasm.
Perfect, yep.
Okay, and the entire job of the optic chiasm is just to basically switch things around and to connect both right fields of vision and to connect both left fields of vision.
I think so. I don't know what else it's used for.
Okay. So both left fields of vision, obviously being the temporal part of your left eye and the nasal part of your right eye, the opposite of what we were talking about before, they would both converge to go back to the right side of the occipital lobe. And I think it's called an optic tract when it leaves the optic chiasm.
It is, exactly.
Okay, so we go...
I don't think they would test you in that, but it's good that you know it.
Well, they could say something like, let's just say that you snip the right optic tract.
Yeah.
What vision would you have left? So if you snipped the right optic tract,
you'd
which
get
would be
no information, right? Because they're all of the
right side of your vision.
Right.
So you would just be in both eyes. You could see the left side, I guess. That's what they would be. That's what they would be looking for. If it was a free response thing, I would say the doctor would be fired. Oops, we snipped your right optic tract.
That's like taking the wrong leg off or something.
Yeah.
Okay. So very, very well done. We even hit how the lens was shaped, which I've seen a couple questions on that, even in the physics section. And so the last two things that we can talk about here we can talk about a lot more with vision, but that's on this AAMC outline would be parallel processing and feature detection.
Ooh, lots of stuff with that.
Yeah, and so I want to talk about feature detection first because it kind of had an aha moment when I read about that one.
I don't know anything about it. I just keep guessing on all of our practice tests.
Yeah, so feature detection is literally what it sounds like. And so it's like the color, the movement, and the shape of whatever you're looking at. And so all of those pieces of information, we have kind of things that tell us those separately.
Okay.
And then parallel processing is adding all of those things together to decide what we're looking at. And so I put in here, it's a bird, it's a plane. Oh, wait, it's the man who wears his bright red underwear on the outside. Oh, wait, Superman. Something like that. But
okay, yeah.
How do we differentiate between each of those three things?
Gotcha.
Right, so
I see here you've written down magnocellular cells and parvocellular cells. Aren't those other ones, like that's with amacrine and all of those, they help us to like really define what we're looking at?
Probably because they're involved in vision. Yeah.
Okay.
Yeah, so the parvocellular cells, color and spatial things, I don't really know how to remember them. I've never learned these before.
Yeah.
But knowing that magnocellular cells means motion or temporal resolution. So at one point in time, someone's right there, and another, like a few seconds later, they're somewhere else. That's temporal resolution. And then cones with our color, we talked about that already. So anyway, feature detection is literally maybe what color is that thing, or what shape is that thing. Like a bird versus a plane will have a different shape versus Superman. So yeah.
All right. And that looks like it's about it for the eye. It's going to be a lot of stuff on psych-soc and a lot of stuff on physics, and probably a lot of stuff on biology too is going to be about the eye. Yeah,
so really know the eye and the ear, which we're about to do another video on. And the last thing I want to mention is that the retina is actually, like Kevin mentioned, the ability to go up and kind of touch the brain through the nose, which I think is how they pulled out brains for mummification in Egypt. Do you remember reading about that?
No.
That's when they put all the organs in those jars, they pulled the brain out through the nose. So anyway, you can really reach it that way. But what I didn't know is that the retina is actually an extension of the brain. How cool is that? Which makes sense, right? It's got things that are nervy all up in there, but how cool.
It's like the brain just spat out your eyes.
Yeah, and like what, 90% of our communication with other humans is physical, right? Body language. So anyway, vision's pretty important, people.
Yeah, strong work, gang.